So I will present a recent work where we automated the operation of an AFM for the purpose of imaging single molecules. And I will put the focus on how we use deep learning techniques for some of the steps involved in this process. Okay, so we are interested in automating the operation of AFMs because, like everyone here, we believe it's a really powerful technique, but it's not probably used as much as one could expect, given its possibilities. Okay, and probably there are two main bottlenecks here, and we have that AFM experiments are really time-consuming most of the times, and you also need highly trained people to do them. And automation could help in this regard. But you can do a lot of things with an AFM, so if we are talking about automation, we have to focus on each type of experiments, and the one that we have focused on this work was imaging single molecules. Okay, so how does one typically perform these experiments? Well, here I wrote a list, and well, basically you scan a large area, you locate where the molecules are, you do progressive zooms on the molecules. We always have drift around until you achieve an image that you consider that has enough resolution, and then maybe you want to perform some kind of force measurement or something similar. And then you zoom out, you localize a different molecule, and you repeat the process until you get statistically significant data. So well, I have spent a lot of my time doing this type of experiments, and I always wanted to reduce this time. So if one wants to automate this type of experiments, what is needed? Okay, well, the first thing is to write some kind of code that will communicate with the AFM or with the software that controls the AFM. And this is something I will not go into it. It will depend on which system you are using, basically. But most commercial setups offer this possibility one way or another. Okay, so, but if you look at this list from a computer vision point of view, there are two steps that are not straightforward. And one is to locate the molecules on the image, okay? And the second one is to be able to differentiate between different molecules. So you don't zoom again and again on the same molecule. And these are basically the two steps for which we use deep learning, and is where I will focus on the talk. Okay, so very brief introduction to deep learning. When we talk about deep learning, we usually talk about multi-layer neural networks. Probably you have seen this drawing many times. And without going into detail, the neural networks are just nonlinear functions, quite complex, but it's just a function that maps some variables into into an output. They are really complex in the sense that they have a lot of parameters that we call weights, but they really work well. They work really well for both regression and classification problems. When you talk about supervised deep learning, this is what we have used for this work. Basically, what you do is to feed this data, to feed these functions. You have a lot of data, and you know the outcome, and yeah, you just do fitting. Of course, you can imagine that when you play with such complex functions, you need a lot of tricks for this fitting. But this is basically what deep learning is. And the point of deep learning is that it works really well with images. If you use what are called convolutional neural networks, where the fitting parameters are basically filters that you apply in an image. And in AFM, or in many AFM experiments, what we have in the end are images. Okay, so if we wanted to automate this type of experiments, well, if you remember the list from the beginning, the first problem we can focus on is how to detect molecules in your image. Okay, in this work I will focus on a model AFM sample as DNA on mica. Okay, so convolutional neural networks are very good for object detection. It's a problem in computer vision that they outperform any other type of algorithm. There are different strategies for algebra detection with neural networks. We have used one that is very fast, and these are the Jolo family networks. I cannot go into detail, but they are really good for embedding systems and in an AFM experiments we want to do things in real time. Okay. So it's basically an algorithm or a neural network that looks, so you have your image is divided into grid cells. Okay, and you get basically two outputs. First, the possibility that each grid is the center of a molecule. So this is a classification output. And you also get the size of the box that will bound the molecule. So it's a regressive output. Okay, so this is these are neural networks that work really well for jet detection. In AFM or for this type of experiments, we had the problem that we want to locate molecules at different sizes. Okay, depending on the area that you are scanning, the molecule will look bigger or smaller. And this is kind of a problem, but we use a specific type of neural network that is called Jolo 3, that is very good at detecting objects at different scales. Basically, you have different outputs where the grid size of the grid cell is different, and then you perform this classification regression problem. So very shortly, this is kind of the output, and they work very well for, in our case, for detecting DNA molecules. You can see that they work at different sizes. Okay. And was no problem to differentiate the molecules from contamination, and also to differentiate molecules that were very close together. There are different metrics that you can use to evaluate the performance of these object detection algorithms, but the conclusion is that it worked pretty well. So we also wanted to differentiate between different molecules. Okay, so this Jolo 3 will locate the different molecules, but then we don't want to focus all the time on the same molecule. And so once we zoom out. So what we did is, after locating the molecules with Jolo 3, we use another type of neural networks, what are called Siamese networks, that are pretty good in this purpose. Okay, so basically, for instance, imagine that first we have focused on this molecule, and then we zoom out, and we want to compare all the molecules detected with Jolo 3 to make sure that we don't focus on the same molecule. Okay, so the output of Jolo 3 goes to this type of structure where we have two convolutional neural networks, actually the same shape and same weights. Okay, and these neural networks embeds the image into some type, for instance, of a dimensional array, and then we find some distance between these two arrays. Okay, so if the distance is low, the two images will contain the same molecule. If the distance is high, it will be a different molecule. And well, this is an example of the performance of these Siamese networks. And well, the distance that we used was just the square Euclidean distance. And you can see that when we input to the Siamese networks images of the same molecule, the distance is quite low and is higher when we input different molecules. Okay, you see again, there are different metrics to evaluate the performance of the network, but you can see that we get all the true positives and almost no false negatives, or not false positive. Okay, so both networks work pretty well for our purpose. So we embedded everything into an algorithm for these experiments, which very briefly work as I mentioned in the beginning. The AFM engage, it scans a large area, locates the different molecules, selects one closest to the center, keeps focusing on this molecule until we reach some threshold that we defined as enough resolution, then zooms out, locates again all the molecules. And if you look, this molecule here is this one, so it knows that it has already focused on this one and selects another one to focus on. Okay, and actually, so it worked pretty well. We could left the AFM running for several days without any user input, and it basically finds out, it takes nice images of all the molecules in your sample, at least all the molecules within the range of your method. And these are examples of images from a single experiment, no user input, and the best resolution images that we got from DNA molecules. Okay, so I think I'm actually on time. I have not gone through technical details, but all of them are in a paper that probably is easy to find online. And the paper also contains the reference to the repository where, if you are interested, you can find all the code and images used for training the neural networks and so on. So thank you very much for your attention.